Hi everyone, my name is William Styers and I'll be presenting my design for a velocity map imaging device that will be used for signal isolation. So let's begin with what velocity map imaging really is. And so velocity map imaging is typically something that uses an electrode stack akin to this stack here on the right, where ion molecules that begin in this general ionization radius who have the same velocity vector will get mapped to the same point in space, akin to this kind of predicted spectra here. And so this is typically used in what's referred to as photodissociation experiments, in which you're using a laser or some other source to actually fragment a molecule and study how that molecule falls apart. However, you can use the same principle of mapping something with the same velocity vector to the same point for other items of interest. In particular, for us, it will be isolating a population of known velocity from background where the velocity is unknown and more chaotic. And so let's look at the system that we'll actually be using this for. It can be seen here on the right. And so one major component is our slit nozzle. This is how we get a molecular beam of gas. And this will be passing through what's referred to as a Stark decelerator chip or Stark guide. What this is able to do is it behaves similar to a quadrupole for a mass spectrometer, only it is an array of them with very small spacing between to produce very high electric fields. And if you, the way a quadrupole is oriented, we have something akin to this where each of these fingers here are electrodes. And we are able to set potentials of each of these electrodes and kind of cycle the potentials around the electrodes such that typically ions passing through it can be retained and separated from each other as it goes on. We are doing the same principle here, only with these stronger fields, we are able to begin and have that effect on neutral species based on only their dipole and polarizability, as well as rotational state. And so we can set the frequency at which that voltage is circulated around the electrodes to select for which molecules will actually make it to the other end, such that only ones with the specific mass to dipole ratio will survive to the other end. And the ones that do escape onto the other end will be exiting all with the same velocity. However, one major problem that we have with this chip is that we are using gas in a vacuum chamber with a very small spacing for the gas to actually enter. And so what that leads to is gas expansion causing most or a large portion of the gas to actually just completely miss our chip. And then that will feed into our detector and make it completely indiscernible from when the chip is actually on and functioning or off and not even being used, which is to say we get no signal, which is a large problem. And so a few of the so that's where velocity map imaging, the stack actually comes in. Molecules exiting this chip will be at the same velocity and completely different from the ones that completely missed it. So should we put some velocity map imaging stack here, we should be able to separate those that were benefited by the chip from those that completely missed the chip. And so a few design considerations that are different from a standard velocity map imaging setup is we're using a planar source of a gas beam that the slit nozzle will produce kind of a plane of gas, whereas most of the time you use almost like a laser beam of gas. And then the molecules that we're actually going to be studying using this are at a much lower energy than molecules typically studied with velocity map imaging by about 10-fold. We're looking at molecules on the order of 0.1 electron volts, while they typically look at molecules on the order of 1 or above electron volts. And so we can see here the design of the stack. We have several lenses, what's referred to as an extractor plate and a repeller plate. Most of these taking on some form of this design here, looking very similar to this. And we can see simulation results of ions that we simulated going through this. And so we have this black body, which would be our background population. Molecules that missed the chip would be considered in this body of population. And then molecules that did not miss this chip will have a different velocity. So as they become ionized and influenced by this repeller plate, they'll begin to separate from this black body. And so we can see here, this green population is molecules of velocity around 600 meters per second and about 0.1 electron volts, while the red population is about 0.07 electron volts. And the black, like I mentioned, is just background noise, molecules that miss the chip. And we can see even between molecules that have around this, the difference in energy produces around 100 meters per second difference in velocity. They produce discernible separation. This here was measured to be around 2.5 millimeters of separation. And we are able to increase the separation as we decrease the repeller plate voltage. However, that kind of smears out the area at which it's mapped to more and more. So it becomes an optimization problem between how refined we want our point that it is being mapped to be versus how separate we want it to be from other populations. And this can be changed for each subsequent run. We can completely change the voltage without having to change the setup or design. And so the methods we did to actually generate this data is we designed the electrodes in a software called Fusion 360, a CAD software. And then we imported them into a program called Simian, which is an ion flight simulator. Within Simian, we were able to write our own Lua code to undergo a simplex optimization routine, which will find ideal voltages to actually apply to each of these electrodes. So we can see ideal voltages that were selected for this run here, with the base plate being 400, the next extractor plate being 363, 327, 290, and 254, with this final plate being a ground plate, always locked at 0. And so the result of it is that we are able to see very good separation between populations just using this chip. We're just using this electrode stack, and we're predicting it will be very good at separating ion or molecules that went through the chip from molecules that did not. In the future, we're working on getting this stack machined and then begin testing experimentally how well it works. And then once we have proof of concept confirmed, we'll begin using it to measure the mass to dipole ratio of many known species of molecules. We may also consider replacing this electron beam with some other ionization source, such as a 2-photon ionization source, where we can be more selective with the molecules we intend to study to separate it from background molecules that are not of that same type. And then currently we're working on actually refining the design to attempt to reduce the cost that it will actually take to machine. Lastly, I'd like to acknowledge the funding from the IRCA grant this past summer. It is the main reason this project has been able to be taken off the ground and worked on. Thank you all for your time.